what we normally do it is even we recommend from the preclinical stage. So you always need to have quality in mind. Okay, I understand in the preclinical you need small amount. You don't need to reach the full scale, but you need also to understand that, okay, the process as it is right now, how it performs, when I scale it up and when I go to the final production scale that I will be producing at, have the same quality. Not when I scale it up, I face some problems and then my host cell DNA increases, my impurities increases, my full empty ratio changes drastically, because then this will not be compliant with regulatories. So you need to keep that early on in mind.
Hey, smart scientists. Welcome to another episode on the Smart Biotech Scientist podcast. Today, we are talking about the development of AAV-based gene therapies. And to do that, I invited Ahmed Youssef, who is a senior manager in the process development team at Ascend, an end-to-end gene therapy development partner. But before we jump into today's episode, please leave a review. Tell us how you like the Smart Biotech Scientist podcast. You can do so by leaving a review on the platform you found us on. Thank you so much for leaving a review. It will help us reach more scientists like you, and more importantly, also to bring topics that are of great value and will help further your career. So we are up for an exciting conversation about the development of AAV-based gene therapies coming up next. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Ahmed, to the Smart Biotech Scientist podcast.
Thanks, David. Really, really glad to be here as well. Thanks for the invite.
Absolutely. It's fantastic to have you on the show today. Ahmed, share something that you believe about bioprocess development that most people disagree with.
I would say there's now a really big abundance in AAV manufacturing capacities. Let's put it like that. And I would say a lot of people would disagree because you've seen previous conferences saying, okay, yep, we do not have manufacturing capacities. We need also to increase the footprint for commercialization. But what I've been seeing is huge growth in the CDMO industry, and a lot of CDMOs are coming in, offering capacities, offering manufacturing capacities, offering slots for GMP production. So there are capacities. The only bottleneck would be actually is having a good, efficient, high quality therapy. Let's put it like that. I'm speaking from the AAV point of view side.
Before we dive into the challenges in getting AAV gene therapies to clinic, let's talk about yourself, Ahmed. So how did you get started in biotech, and what pivotal moments and decisions brought you to your current role?
I am a pharmacist by education. So basically, I graduated with a bachelor in pharmacy and a minor in biotechnology, where the field was still evolving at that time in 2005. So I said, okay, was really interesting to delve more into this molecular biology part, into these new therapy modalities. Monoclonal antibodies was rising at that time. For me, I said, okay, I have traditional pharmaceutics and I have the new generation of biotech. So that was what really drove me to go more towards that area and further continuing my studies, taking my master's there, and then working in the industry where I can really apply that into tangible products to help patients' lives.
And since you've seen various parts of the industry, I'm curious, Ahmed, What do you think are the main challenges that biotech scientists and engineers often encounter in bioprocess development and the manufacturing of the biologics?
I would say biotech scientists just fresh out of their graduate studies, for example, joining in the industry, sometimes they have a lot of really, really great ideas, but not really translatable or can be transferred into an industrial commercialization setting, needs some still development into a more probable solution to fit in the industry and then address a certain issue, which I mean is the base of a lot of advancements that happened in the biotech industry as well. But they come really with a very good passion, and they come really with a lot of energy to put into that process to learn and to contribute to that with their ideas. And this is what keeps the industry moving forward.
Now, in your current role, Ahmed, you are in the AAV space. So, to start our conversation about this fascinating topic, let's talk about the challenges in getting AAV based gene therapies to the clinics. Can you tell us more about that?
So, challenges to go to the clinic is, I would say, really, really focused about the quality. So, a lot of companies, a lot of startups, they have a very, very good product. They have a very good idea that they want to put, but they want to raise to get funding, to get more resources, to build up their company, to build up their capabilities. So they can, I don't want to say take shortcuts, but they do not really focus into the quality at earlier stages, which for them, when they're doing, preclinical studies, some small animal studies, they do not focus into say, okay, the material is good enough to do that. But when you really then scale it up and go to clinics and phase 3 trials, you get the scrutiny from the regulators that you need more and more in terms of quality, more and more in terms of the characterization of your process. Then they look into that, then let's do it right now. Doing things right at a later stage is a very, very costly endeavor. And that's, I would say, this is one what we call it the stumbling blocks in CMC development, when you reach at the end and then you need really to reinvest a lot, generate a lot of materials early on, do the process right. And in some cases, regulatories force you to repeat some of the clinical studies as well, even if smaller groups, but just to prove comparability of this new version of the process versus what you previously produced. I don't want to say in a quick and dirty manner, but just to get things moving along. So really, I sometimes feel that for the non-educated, quality is overseen, and it's a very, very critical point in the AAV. I would say it's impacts the development of a lot of products that could have been commercialized in the market. If you pay attention to that early on, when you start your clinical phases, start to talk to regulators to get their insights, are you moving in the right directions or not? They're always tightening their specifications for impurities. You need to keep an eye on that. So having all of these considered would make your process to launch your product, commercialize it much more smoother and scalable and reliable, from the small scale you do for animal studies to the larger scale you do for the clinical trials.
When you say early on, Ahmed, when is that? Is that before moving into clinics? Is that phase 1? Or what is your recommendation?
Even before phase 1. So what we normally do it is even we recommend from the preclinical stage. So you always need to have quality in mind. Okay, I understand in the preclinical you need a small amount. You don't need to reach the full scale, but you need also to understand that, okay, the process as it is right now, how it performs, when I scale it up and when I go to the final production scale that I will be producing at, have the same quality. Not when I scale it up, I face some problems and then my host cell DNA increases, my impurities increases, my full empty ratio changes drastically, because then this will not be compliant with regulatories. So you need to keep that early on in mind.
Yeah, smart biotech scientists, this is excellent advice. Keep the quality in mind from the very beginning. It will save you a lot of time and a lot of money. Can you walk us through the typical steps of an AAV manufacturing process? Because I'm sure certain of our listeners, they are not familiar with AAV. So how do you produce these?
So we produce AAV based on a transit transfection with HEC293 cells. So one of the most common, I would say, host cell lines where you do that. There are different modalities where you have stable producer cell lines or cell lines for transit transfection, like what we do. So in essence, at the end of the day, you reach the AAV molecule that you want. But let's take it from the early on. You have a cell, you have plasmids that encode the capsid genes, so the specific serotype that you want your AAV basically molecule to be, and also you have the gene of interest that basically is your therapeutic gene of interest that you want to incorporate. So whether you are using the traditional three plasmid system or whether you're using a more superior split plasmid system, you do the transfection, it transiently introduce that to the cells, and then the cells start to produce your viruses. Under normal conditions, the viruses are sequestered within the cells because they are not released outside, except for specific serotypes like AAV8, which is more prone to the viruses to be secreted outside, which is one also of the very big differentiators between when you produce a biomolecule like monoclonal antibodies versus AAV. Monoclonal antibodies, for example, are they have the tendency from CHO cells to be secreted outside. You have a lot of options to just collect them, do continuous processing, continuous DSP. But for AAV, you always need to introduce a step where you lyse the cells to release the AAV outside, unless you have a genetically modified cell line that is doing that for you. Once you lyse the cells, you have now all your, let's say, bulk harvest, where you need them to clarify that, clean all the cell debris, clean all these contaminants, make it in a more, not purified, in a more clarified format to be fitting to go into your DSP. And from there, DSP takes over to do the purification. And again, there are multiple modalities where people concentrate their bulk harvest first to very small volume that they can process, or based now on the new technologies available and the resins with the higher capacities, you can just load all of everything on the affinity chromatography and start your purification early on, where you then focus into removing everything contaminating your AAV preparation, which is non-encapsulated, which is outside your capsid, like host cell protein, anything that is from the outside, you just leave your AAV pure. You still have impurities inside your AAV itself, which are encapsulated, which are mainly affected by your upstream process, which then you play around with the plasmid ratios, the plasmid amounts, the USP conditions, the steering, oxygenation, pH, all of these parameters to try to get the best high quality AAV that you can produce.
And what is the specific advantage of HEC-293 cells for AAV production? Why are we using these and not other cell lines?
These cell lines are widely used because they are prone or susceptible to the transfection and transient transfection in this case. So they can easily be transfected, and then you can take that and then they can easily be manipulated to produce your AAVs. There is a lot of work ongoing as well in the industry with other cell lines, but I would say this is the majority of the cell line everyone uses for the ease of use and easy to be manipulated.
So now when you develop a process, you have to look at both the process performance and then the product you're producing. So the quality of the product and how efficacious that will be then once a patient is being treated. So can you tell us what are the key considerations we should keep in mind when designing a process?
So I would say you really hit a very, very nice point, David, here. And it comes back to my original point, which we're talking about quality. Early on, you need to define what we call target product profile. So this is basically your quality profile, the boundaries that you want to put your molecule in. And this is also you need to keep at each scale and at each stage, whether it's a preclinical or clinical or commercial stage. So you say, okay, my target profile, for example, would say I would have host cell DNA impurities of less than 10 nanograms per 1E12 viral genome dose, for example. Or I would be having plasmid-derived impurities in total less than 10%. So you put the boundaries of your process, and these boundaries, you help set them up with the guidelines you have also from the regulators, where they say, okay, we want to ensure final material does not have impurities more than that or less than that, or full AAV particles have certain limit. Let's say, okay, we need minimum 70% full, just another example, or minimum 50% full AAV particles. You really need to keep quality in mind, have a lot of analytical tools to be able to understand what's packaged inside your AAV and how does that impact the performance. Coming to your point with efficacy, you would always also try to test that in vitro, like using cell-based assays, where you say, okay, I will be testing potency. For example, in my company, we try a mixture of a lot of things. We try a mixture of PCR-based methods to quantify the impurities inside the capsids. Then we merge that with large read next generation sequencing to be able to sequence what's inside and match that with the PCR data we get so that we know, okay, are we packaging really actual gene of interest, or are we packaging junk, or are we packaging host cell DNA packaged inside, which at the end of the day might be particles are not efficient. And you also need to check how many particles I have they are empty, how many particles I have that have something inside, and what is that thing? Is it efficacious or is just truncated sequences? And with that, you augment that finally with testing the potency and checking and then saying, okay, is the potency of my preparation okay? Is the potency once I have it at the end of the USP the same as I finish with the DSP, or did the DSP introduce something that decreased the potency? Or the other way around, is the potency from one preparation to the other consistent, or from one scale to the other consistent or not? So having all of that package give you more understanding and characterization of your AAV preparation, and at the same time facilitates the discussions with the regulators. You understand your product, you know the qualities, that limits that you have put to yourself, and you know how to keep that in line all over the different scales.
Now, how do we optimize the vector production? Because yes, we don't want to have empty capsids. We want to have the right quality and actually the most, if possible. What are the key strategies we can apply?
I would say two key strategies. One would be the cell line itself, so that the better the cell line you have, the better quality you will be getting for the AAV. Better packaging, cells are less stressed, so you have better efficiency of what you're getting more viral particles with, let's say, the good packaged gene of interest inside rather than having empties or junk. In addition to having a very good plasmid system that is meticulously developed so that you do not, for example, you prevent any events of any recombination or replication competent AAV generation, or you also prevent any event that you could have plasmid-derived impurities under the influence of a promoter that then could be detrimental, in this case, if they are inside an AAV molecule. So you try with the plasmid system that you have in conjunction with the cells, in conjunction with the transient transfection conditions that you're doing. I would say the DNA to the transfection reagent ratio. The transfection reagent itself, there's a lot of transfection reagents in the market. They have the pros and cons. From that, some of them we've tested in my group produced very, very good. So you have very, very high titers, but you also have a very bad quality. So you need to balance really the yield and the quality, again, to fit your target product profile, but also make the process economical and commercial to be able to feasibly commercialize it. Don't focus only on the yield, saying, okay, I reached these limits, because then the quality could be very bad, potency could be very bad, full particles could also be very bad. Then you will need at the end to say, okay, I need to insert a step in the DSP where I have full enrichment. So I do the full empty separation, what they call, or a full enrichment step, which overall would decrease some of your yield, add costs, add more complexity to your process. But if you define or use a very good plasmid system, very good platform that at the end would be really showing that you have very high level of full particles, very good quality early on, this will facilitate your DSP, and then your DSP will be seamlessly easy and smooth.
Hey, smart biotech scientists. I hope you've been enjoying our conversation with Ahmed Youssef, who is a senior manager in the process development team at Ascent. What did you think about today's episode? Please tell us by leaving a review on Apple Podcasts or whatever platform you found us on. And we are continuing our conversation about the development of AAV gene therapies on Thursday. And perhaps you are developing novel therapies yourself and you're not so sure how to do that well. Well, we have developed a fractional CTO service to come in and partner with you and help you develop a solid strategy and then help you execute this strategy. If this sounds interesting, I want to offer you a free 30-minute assessment to go through your specific requirements and to help you get started and to help you see whether this program, this service, this fractional CTO service would be a good fit in order to help you develop your novel therapies. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.